Okay, can somebody confirm you can hear me?
I can hear you.
Thank
I
you.
can hear you, doctor.
Thank you. Let me share my screen. So today is April 25th, and we are supposed to be on chapter 3, observing microorganisms. There is no lab today. We did lab 4 on Tuesday. I will be logging in during the lab time for the first 15 minutes to answer any questions. If there's no further questions after 15 minutes, I will log out. Any questions about what we're doing? All right. Which allows me to select where we left off. Where do I have the find? There it is. All right, I have that we were talking about staining microbial smears. Is that correct? Is that where we left off?
Yes.
Thank you. So the purpose of a stain is simply, or a dye, is simply to color the specimen or the specimen's background and then make the specimen have higher contrast with the background of the slide. And we do that with dyes or stains, which are salts consisting of a positive or negative ion, one of them being colored, and that we call the chromophore. If the chromophore is in the positive portion of the salt, then it is a basic dye. But if the colored portion or the chromophore is in the negative portion of the salt, it is an acidic dye. Bacteria have a slightly negative charge, and therefore a basic dye is better for staining the cell. Some basic dyes include crystal violet, methylene blue, malachite green, and safranin. And these are the commonly used stains in microbiology. Acidic dyes normally stain the background, not the microbe. Some examples of an acidic dye are eosin, acid fuchsin, and nigrosin. If you stain the background instead of the cell, it is called negative staining. Any question about that? So an acidic dye is often called a negative stain. Any questions? The term negative comes from the old Polaroid film. You used to take a picture with a Polaroid camera and it would give you a negative and everything would be highlighted in black and white, but in the opposite what you expect. So normally, like if someone was wearing dark clothes, their face would be light in the picture and the clothes would be dark. Well, the negative would be the opposite, where the face would be dark and then the dark clothes would be light in the negative. Any question about any of that? I imagine most of you have never seen a negative for Polaroid film. Is that correct? All right,
I've seen a negative before.
you've seen a negative, great. I said most of you, not all of you. But when we stain using a basic stain, the cells will look like this, where the stains are darkly stained by the cell. And in this case, the background is lightly stained. Usually the background isn't stained at all with a basic stain. And then with a negative stain or an acid stain, it will stain the background darkly, and then the cells are not stained. Either stain, the acid stain or the basic stain, will show you very well the cells. Any question about any of that? This is a simple stain because we're only using one stain, and we'll talk a little bit more about that. There are three main types of stains: a simple stain, a differential stain, and then special stains. A simple stain is an aqueous or alcohol solution containing a single dye. Its primary purpose is simply to increase the contrast between the specimen and the background of the slide. The stain is applied, and you usually apply it to a fixed smear, meaning a fixed bacterial smear if you're staining bacteria, for a certain length of time. The stain is then dropped off, meaning you take the slide and you drop the stain off. And most of the time you put that in a special place. You don't put it down the sink unless it's a non-toxic stain. And then you usually rinse it off. It's then allowed to dry, and then you examine it. So to do the stain, you usually have a bacterial smear made before you do the staining process. Any question about any of that? A mordant is not a simple stain, but it may be used to intensify the stain. We'll talk about a mordant when we're talking about the Gram stain. Gram's iodine is a mordant. It combines with the first stain and then intensifies the stain. It usually increases the affinity between the stain and the specimen. It may also coat the specimen to enhance it, thereby making it easier to see by increasing the size of it, really, because you're putting down several layers on the specimen. An example of the latter is the flagella stain, where you're coating the specimen and you're putting on many layers on the flagella, and now the flagella can actually be seen in the flagella stain because the flagella is now stained and you can see it with your eyes under the microscope. Whereas where you're to do another stain, like a simple stain, the flagella will not stain because it just won't be enough for the human eye to see. You would be able to see it under the electron microscope, but under the light microscope, the human eye would not be able to see it unless you coat it many times with the flagella stain, in which case the flagella can be seen, as shown here. The differential stain reacts differently with different kinds of microbes. So if you have different species and you use a differential stain, the stain can stain differently, and then you can distinguish the two cells by their differences. Usually they have a different color. Usually two dyes are used in a differential stain, but that is not required. The most commonly used differential stains in microbiology are the Gram stain and the acid-fast stain. Any questions about any of that? The Gram stain was developed in 1884 by Hans Christian Gram because it's named after a person. Whenever you use the Gram stain, the G should be capitalized. Any question about any of that? Every term students get that wrong. And I don't mark you down there, but you should capitalize the G whenever you're talking about the Gram stain. The Gram stain turned out to be a very useful stain in bacteriology because it classified bacteria into two very large groups, the Gram-positive cells and the Gram-negative cells. The Gram-positive cells stain purple. The Gram-negative cells stain pinkish under the Gram stain. And they do have differences. There are differences in the size of their and the thickness of their cell wall. All right. Any questions about any of that so far? I haven't talked about the special stains. Let me see. I will at the very end. The special stain, what it does is it stains a particular part of the cell. And then that allows you to see that part of the cell better. Like maybe it's the flagella stain. That's a stain that stains the flagella. And that's only a cell structure. So we call it a special stain. If there's no further questions, let's talk a little bit further about the Gram stain. We're going to talk about this stain in depth. Here's an overview of the Gram stain. Before you employ the Gram staining procedure, you first make a bacterial smear. And you have the bacterial smear on the glass slide. Normally, what you do when you make a bacterial smear is you put a drop of water on the slide. You then add the bacteria to the smear the drop and the bacteria all over the slide, let it air dry. Then you heat fix the bacteria onto the slide. The air drying and the heat fixing attaches the bacteria to the slide so the bacteria will not be washed off in the staining procedure. So you make the bacterial smear first. It is a separate procedure from staining the cell. Any questions about any of that? Because every term I have a student who gets something wrong on that. The bacteria smear, making the bacteria smear, is not staining the cell. You then start the stain of the cell. And in the Gram stain, what we do is we first flood the cells with crystal violet. We apply it for 1 minute. After 1 minute, you dump the crystal violet off, and then you wash with cold water. You then apply iodine called Gram's iodine. It's a mordant. You apply it for 1 minute. You then wash that off with cold water. You then flood with acetone ethanol. This is the decolorizing agent. You only apply the decolorizing agent for about 5 seconds. You then wash immediately the decolorizing agent off of the cells. You then flood the slide with safranin. Safranin is the second stain, so we call it the counter stain. You apply for 1 to 2 minutes. You then wash the safranin off, and then you blot dry and you examine the cells under the microscope. Any question on the basic procedure? We'll talk about the in-depth steps in just a minute. So in the Gram stain, when you first apply the crystal violet, let me blow this up a little. All of the stains cells will take up the crystal violet and they will be a bluish purple color. You then apply the Gram's iodine and they will be a purple color, meaning the color will change a little different. It'll be a little less blue and a little more purple in color. But it'll also be a little bit more intense. The reason is because the Gram's iodine complexes with the crystal violet, making a larger molecule or complex in the cell. And so the cell will show up a little bit more intensely. You have to have a really keen eye to notice that. But the color is slightly different too. It's a little more purple. You then wash that off and add the decolorizing agent, which in this case is an alcohol wash. The decolorizing agent will pull the crystal violet iodine complex out of the gram-negative cells, but not out of the gram-positive cells. And this is assuming that we're doing the gram stain correctly. We'll talk more about that. You then immediately wash the decolorizing agent out so it'll stop working. And then you apply safranin, and safranin will get in the cells and color the clear cells after the decolorizing step, meaning the gram-negative cells, will be stained pink by the safranin. Any question about that? And the gram-positive cells retain the crystal violet iodine complex, so they continue to be purple. You will need to know the different steps and the different colors of the cells at each step in the gram stain. All right, so in the gram staining procedure in depth, we have a air dried and heat fixed bacterial smear. We take that bacterial smear and now we begin the gram stain. We flood the slide with crystal violet. It's the primary stain because it's the first dye used and it colors all of the cells the same color. You apply the crystal violet for 1 minute. Crystal violet sticks to the peptidoglycan in the cell wall. And then after 1 minute, you dump the crystal violet off and then wash the cells in cold water to remove the trace crystal violet. It doesn't remove it from the cells, but it does remove it like from the slide. After applying the crystal violet, all the cells will be a blue-purple color. I mean, the gram-positive will be blue-purple. The gram-negative will be blue-purple. You then apply iodine. Grams iodine is a mordant. It increases the affinity for the crystal violet. For the specimen, the grams iodine actually complexes with the crystal violet, making a larger molecule. You apply the grams iodine for 1 minute, and then you dump it off and then wash gently with cold water. Once again, after the application of the mordant, the color of the gram positive and gram negative cells will be the same. It'll be a purple color, meaning the blue purple has changed to a little more purple color. If you were to look carefully, you would notice the cells will be staining a little more intensely. And that's just because the iodine is a larger molecule than the iodine crystal violet complex is a little larger molecule than the crystal violet itself. You then apply the decolorizing agent. There are several decolorizing agents in this slide, and the decolorizing agent that we use in the microbiology lab the most is acetone ethanol. It washes the crystal violet iodine complex out of the gram-negative cells, but not out of the gram-positive cells, as long as you do not over-decolorize. It is considered the differential step in the gram stain because the gram-positive cells will stain differentially from the gram-negative cells after you apply the decolorizing agent. Any question about that? So with the application of the decolorizing agent, you apply it only for about 5 minutes. Hopefully you can see this if I put it over my face. The best way to use the decolorizing agent is to get your slide, get it where you can see it, meaning you're looking at it, and then apply the decolorizing agent at the top of the slide so it will run down the slide. And as you're applying the decolorizing agent, once the purple stops running on the slide, stop applying the decolorizing agent. If you apply the decolorizing agent for too long, you will pull out the crystal violet iodine complex from the gram-positive cells, and you don't want that to happen. So after about 5 seconds, and like I said, the best thing to do is just to apply it, and then when the purple stops running, stop applying it. You then wash the slide immediately because the trace agents that are remaining on the slide will continue to work and will, if given enough time, pull out the crystal violet iodine complex from the gram-positive cells. We don't want that to happen, so you wash the slide immediately to remove the decolorizing agent. After the application of the decolorizing agent, the gram-positive cells will be staining purple and the gram-negative cells will be colorless. Any question about any of that? And this is why we call this the differential step, because the cells have a different color after the step. You then apply safranin. It's a pink dye. It does stain all bacteria. It's this counter stain because it's the second dye we're using, and it has a different color from the primary stain, which in this case is crystal violet. You flood the safranin for 1 to 2 minutes. At Clark College, I've noticed the safranin is always strong enough that 1 minute is sufficient. But I've taught at Portland Community College, and some terms they made it a little too weak, and you needed to apply the safranin for 2 minutes. But at Clark College, it's always made strong enough the way it should be, and 1 minute is sufficient. You then dump the safranin off and wash the slide in gently with cold water. Any question about any of that? After the application of the counter stain, meaning the safranin, the gram-positive cells will stain purple, and the gram-negative cells will stain pink. You then blot dry the slide in bibulous paper. It's kind of thick paper. I don't have thick paper, but I'll try and show you. You fold over the bibulous paper, put the slide inside, and then blot it dry. You just press the bibulous paper onto the slide. You do not move the bibulous paper against the slide like that, because that'll remove the cells. You just want to remove the majority of the water, and enough water will be removed that you can then look at the slide under the microscope, and you may even apply oil. Enough water has been removed to apply oil to the slide. Any question about any of that? All right, before we go here, let me talk about something. Remember we're on slide 43 here. I forgot to say this at the very start. There we go. After every step, we're washing the cells with cold water, washing them gently so we don't wash the cells off the slide. But after every step, you're washing with cold water. When you're detailing the gram stain, you do need to state that you flood this slide with crystal violet, and then you flood with grams iodine. You then apply the decolorizing agent. All those steps need to be on there. However, what you can do instead of putting in after each step, you're washing with cold water, you can just state it one time and then state something like, and we apply the cold water wash after each step of the gram stain. Okay, so you can do that at the start or at the end, but you don't need to put in after each step this cold water wash if ever you're detailing the gram stain. Okay, and there's only one time you need to wash immediately, and that's after you add the decolorizing agent. All the other times you can let the slide sit. It's not going to harm the slide as long as the slide doesn't air dry. So you do need to, I guess, wash with the cold water within, I don't know, about 5 minutes, maybe within 2 minutes if it's a real hot day. But the point is, it doesn't need to be immediate. All right, what slide were we on? Was that 43?
Yes.
All right. So the basis of the gram staining, the gram positive cells are purple after the procedure. The gram negative cells are pink after the procedure. Why? What is the basis of the differential staining? It's related to the differences in the cell wall. They have different structures. The gram-positive bacteria have a thick cell wall. And in bacteria, it's a thick layer of peptidoglycan. We'll talk more about peptidoglycan in a little bit or in the next lesson. I don't remember which. It's a disaccharide and has some short peptides in it, so disaccharides and amino acids. And it makes that cell wall of the bacteria very thick. The gram-negative bacteria do have peptidoglycan in their cell wall, but it's a much thinner layer of peptidoglycan. And then the gram-negative cells also have a lipopolysaccharide layer outside of the peptidoglycan. So their cell wall is very different. Any question about any of that? So here we're looking at the gram-positive cell. As you can see, a very thick cell wall shown in purple. On the gram-negative cell, let me see if I can blow that up. On the gram-negative cell, we have a thinner peptidoglycan layer, which is shown in purple. And then we have a yellow layer outside of it, which is a lipid bilayer, similar to the cell membrane, which is the lipid bilayer there. And the gram-negative cell, of course, has that lipid bilayer because it's the cell membrane. So the cell membrane. And then there's the cell wall on the gram-positive cell. On the gram-negative cell, we have the peptidoglycan layer, but we also have a lipid bilayer outside of it, which is part of the cell wall. On the, let me see if I can blow this up. There we go. On the gram-positive cell, we have layer upon layer of peptidoglycan. There are some other molecules in it, but the main molecule is peptidoglycan outside of the plasma membrane. And this slide is only showing you 3 layers of peptidoglycan. In reality, it's between over 20 and over 100 layers of peptidoglycan. Okay. On the gram-negative cell, there's the cell membrane. it's showing you the layer of peptidoglycan. And in this slide, it's only showing you one. In reality, in the gram-negative cell, there are at least 3 layers of peptidoglycan and maybe up to 20 layers of peptidoglycan. And then outside the peptidoglycan, there is a lipid bilayer similar to the plasma membrane that we call the lipopolysaccharide, and we call it another name. It's called the outer membrane of the cell wall. So it is a part of the cell wall, but it is an outer membrane on the outside of the cell wall. Any question about any of that? All right, the crystal violet iodine complex forms in all cells, both the gram-positive and gram-negative, after the addition of the grams iodine. The crystal violet iodine complex is larger than the crystal violet or iodine alone, so it intensifies as a stain. During the alcohol acetone wash, meaning the decolorizing step, the gram-negative cells have their crystal violet iodine complex washed out because the peptidoglycan layer is thin. And the lipopolysaccharide layer is actually disrupted by the alcohol wash, meaning it's sort of dissolved by the alcohol. And so the crystal violet iodine is washed out and the cells will become colorless. The gram-positive cells, on the other hand, the crystal violet iodine cannot be washed out because the thick, intact peptidoglycan layer retains the crystal violet iodine complex. So as long as you do not over decolorize, the gram-positive cells will remain purple after the decolorizing step. Any question about any of that? So here we see in the gram stain that we have different cells here, and there's gram-positive cells and gram-negative cells. The gram-positive cells are the purple staining cells. There we have purple bacilli. Here we have purple cocci. There we have a purple rod or a bacilli. There we have a purple spirilla. There's another purple spirilla. There's a purple vibrio. We'll talk a little bit about that if I haven't talked about that. Any questions on the gram stain? When you apply the gram stain, you will have some, I don't know, problems with it. Like when you get a smear that looks like this, what do you do for determining the color? Like those look kind of pink right there, and these in this area are definitely dark purple. Which cells do you read? And do you even look at the cells in this region? These are bad staining regions. That actually is not cells. What those are is a crystal of crystal violet. And what happens is when you make crystal violet, sometimes the crystal violet will start precipitating out of solution, particularly if you have an old bottle of crystal violet. And then if you apply from the bottom of the container of crystal violet, or it just gets mixed up and then some of the crystals are in solution, and then you pull from the top, you can get the crystals, and then you apply it to the slide. The crystal will just precipitate, it's already precipitated, and then just go on the slide, and it'll do that. And if you look closely, let me see if I can blow this up. You'll note that these cells really close to the crystal are darker staining than the cells further out. So it's like the crystal violet came off of the crystal and then stained the nearby cells a little darker. than the cells further away. So you should not look at the slide in the bad regions where there are crystals of crystal violet. You should look at the slides in the good region where the crystals are not and where the cells are also in a single layer thick. Any question about any of that? So when you actually were to do the gram stain, which I guess you never will because everything we're going to be doing will be a virtual, meaning you might do the gram stain in the virtual lab, but beyond that, you won't be in a lab to do the gram stain. But it does have some, I don't know, hair pulling, some problems you need to work with. And that's one. Some regions are good, other regions are not so good. And then some other hair pulling with the gram stain. When you apply the gram stain, you should only apply it to freshly grown cells. Bacteria that are over 2 days old may not stain correctly in the gram stain. So if you don't have cells that are 2 days old or younger, then you should grow up some cells and then only use cells that are at most 2 days old. The gram stain is not universally applicable. Some bacteria cells stain poorly or not at all. It's usually the mycobacteria that stain poorly. And that's because they have mycolic acid in their cell wall. Mycolic acid resists water or anything dissolved in water, like the crystal violet. And so the cells, they usually stain lightly, meaning kind of poorly, but they do stain. At least all of the mycobacterium I've looked at. But the point is, the results are not always as clear-cut as the theory suggests. And when you do the unknown project, if you were to do the gram stain, that would become very evident to you, where you start pulling out your hair because the gram stain didn't quite work the way it should have. The structure of the gram positive and gram negative cell walls not only affects the staining, it's also related to how well the cells respond to certain antibiotics and to other treatments. For example, the gram-positive bacteria tend to be easily killed by penicillin G, as well as cephalosporin and many detergents. It's because penicillin G, cephalosporin, and many detergents are water soluble. And these agents get right through the cell wall. But the gram-negative bacteria are more resistant to these antibiotics, meaning penicillin G, cephalosporin, and some detergents, because they have an outer membrane of the cell wall, which blocks penicillin G from getting into the cell. And it can block cephalosporin and many detergents from getting into the cell. If it can't get into the cell, it can't damage the cell. And so gram-negative bacteria are resistant to penicillin G, as well as cephalosporin and some detergents. Any question about that? Now, that doesn't mean that gram-negative cells are more resistant than gram-positive cells to antibiotic treatment. You just need to use the correct antibiotic. For example, ampicillin, one of the earlier members that was developed in the penicillin family, works on both gram-negative and gram-positive cells. So it can get through the outer membrane of the gram-negative cell wall. And it can kill gram-negative as well as gram-positive cells. Penicillin G, on the other hand, tends to only kill gram-positive cells. All right, any questions about any of that? Any questions about the gram stain in general? If not, let's just talk a little bit about special stains. You're not going to have a quiz question on this other than the definition for what a special stain is. A stain that's used to stain or color a specific part of the cell. But I do need to talk about the capsule stain because some of you may use it in your unknown project to stain the capsule of a bacteria. A capsule is a secretion of the cell that surrounds the cell. And when you apply the capsule stain, you will note that the capsule does not stain. So there's the bacteria, very dark color, very dark purple, and the background of the cell stains a lighter purple, and then the capsule is this halo, an unstaining region around the cell. Here's another example of the capsule stain. This is India ink. It's a negative stain because we're just applying the India ink and leaving it on the slide and then looking at it under the microscope where the cells are still wet and the India ink is still wet as well. And the India ink stains the background of the cells very dark, almost a black color. It does stain the cells lightly, a gray color, and then the capsule is unstained. Any question about special stains? All right, if not, let me see. Let me begin chapter 4, but we won't go very far, and then we will end the lesson early today. Any questions about chapter 3? If not, let me save that down. Hopefully I remember to remove the 2 here. As is normal, I show you a introductory slide listing the major goals and the rough outline of the lesson. So chapter 4 is the functional anatomy of prokaryote and eukaryote cells. Let me warn you that if we have time, which I almost never do, we will talk about the anatomy of prokaryotic cells. But eukaryotic cells, you should have learned in Biology 160. So if I run out of time, I'm not going to cover it in this lesson. And then we'll move on to the next lesson, chapter 5. But you will be held responsible because it's something that you should be just review for you. And I do have the information in my slide here. That reminds me, let me go to lessons and make chapter 3 available for you. Hopefully I made chapter 2 available already. Yep. So always know the terms. What's a coccus, a bacillus, a spirilla? These are different shapes of bacteria. What's a streptococci? What is a staphylococci, meaning what's strepto something, staphylo, diplo, tetrad, what's a sarxinia, what's a vibrio, what's a spirochete, what's the glycocalyx, what's a flagella, axial filaments, a fimbriae, a pili, peptidoglycan, we've already talked about that one. Endotoxin, what's an endotoxin? What is selective impermeability? What's diffusion? Facilitated diffusion, osmosis. What is active transport? Tonicity. What is a plasmid? What are inclusions? Endospores. And lastly, we will talk about the endosymbiotic theory. The endosymbiotic theory is something that is in the cover the section with the eukaryotic cell. You probably never had the endosymbiotic theory, so I will cover that when we're talking about eukaryotic cells. And it may be the only thing we talk about with eukaryotic cells. And then 2, explain how gram-positive and gram-negative cell walls differ. And then 3, describe the importance of taxis in bacteria. Any question about what we're going to cover? So in prokaryotes, the word or term prokaryotes is actually Greek for pre-nucleus. Literally, it translates to pre-kernel, but they're using kernel for nucleus. And eukaryote is a Greek word for meaning true nucleus. The prokaryotes, or at least bacteria, has 1 circular chromosome. The bacteria, the DNA is not in a nucleus. The eukaryotes have linear chromosomes, and the bacteria are in a nucleus. Let me shut down the mail. So I have to get into my mail to start the course. That's why that was open. So we don't have any other dings like that. Not that that's terribly important, but where are we? Now, when we said the chromosome is circular, it means the chromosome is in a circle. So trying to draw a circle here, maybe a big one like there. A circle has the beginning point where the ending point is. So it's a circle. The eukaryotes have their chromosomes like the X chromosome in a, oh, I can't draw it here, never mind. In an X, like the X chromosome. Where is, that's weird. Normally get paint to gum with that. I don't see paint here. Let me put it in. There we go. So the X chromosome has a beginning point right here and an ending point right there, which these are not the same point. Whereas in a circle, your ending point is the same point as your beginning point. Any questions about any of that? And of course, the chromosomes, usually there's several of them or many of them, they're in the nucleus. The DNA of prokaryotes, especially bacteria, is not associated with histone proteins. The DNA of eukaryotes is associated with histones. So the main protein in the chromosome of eukaryotes is a histone protein, which is a real big protein. And the DNA actually wraps around the histone protein twice in a eukaryote chromosome. The bacterial chromosome does have proteins in it, but it does not have histone protein. There are no membrane-bound organelles in any prokaryote, and there are membrane-bound organelles in eukaryotes. In bacteria, there is peptidoglycan in their cell wall. If they have a cell wall, they have peptidoglycan in it. In eukaryotes, there is no peptidoglycan in the cell wall. And you'll note I mentioned in bacteria only. The archaea do not have peptidoglycan. The prokaryotes divide mainly by binary fission, where the cell simply splits into two. Eukaryotes, their cells divide by mitosis, which is a topic you should have had in Biology 160. So in prokaryotes, we're going to talk about the cell shape, talk about their cellular arrangement, also called the cellular growth arrangement. We'll then move on to cell structures, move on to movement of material across the membranes, especially the cell membrane. And that's pretty much it for prokaryotes. If we have time, which I imagine we will not, we will then talk about the cell structures of eukaryotes. And then lastly, I will talk about the endosymbiotic theory. It's actually the theory of how eukaryote cells came to be. Any question about any of that? All right. When we're talking about prokaryote sizes and shapes, they come in different sizes and shape, with the average bacteria being about 1 to 2 micrometers in length. A 10 would be a very large bacteria. I don't remember if that's the largest bacteria or not. The smallest is actually smaller than 1 micrometer. They think the largest viable bacteria is about 0.4 or 0.4 micrometers in length. But there are non-viable bacteria, which are a little smaller, and they can go down to 0.2 micrometers in length. They have different shapes, but they have 3 main shapes. So bacteria come mainly in 3 main shapes. A coccus, which is a round ball, shown here. A bacillus, which is a rod, shown here. And then a spiral, which is an S-type shape, shown here. And there are different shapes in bacteria, but most of them are some combination of these 3 shapes. Specifically, the coccobacillus is a very short bacilli, which has rounded edges. And as you shrink it, you have something in between a bacillus and a coccus, meaning you have an oblong coccus, or you have a, I don't know, a round bacillus, something like that. Roundish bacillus. And that's a coccobacillus, a shape that is exactly between a bacillus and a coccus. Any question about that? And then there's a spirochete, which is a spiral, but it has many spirals in it. And we'll talk a little bit more about that. I'll show you that. Maybe not today. There's a spirochete right there. So there's the normal spiral, and there's the spirochete. It has many spirals in it. There's actually some differences between the spirals and the spirochetes, and that is the spirillums are more rigid in their shape, where the spirochete is more flexible. This cell can actually, I don't know how to do that, show you with some paper. So pretend this is the spirochete. It can actually change its shape and fold up like that. And it usually does that, like when it's moving this way, it wants to move north. It usually go like this, and then go like that, and then start moving that way. So my point is it's more flexible than the spirals. And then there are a different type of a spiral, and you can call this a half of a spiral or a bent rod. So a vibrio is a curved rod or a half of a spiral. Okay? You can call the vibrio either way. There are some bacteria with unusual shapes, like the Stella bacteria actually has a star-like shape. And that's an actual electron microscopic image of the Stella bacteria. And then the Haloarcula of Archaea is a squarish shape. Artist picture, that's the actual shape there. And actually, it's not a square, it's a rectangle. Any question about that? So some bacteria can have unusual shapes, but most of them are either a coccus, a bacillus, or a spiral, or some variation of that. Most bacteria species are monomorphic, meaning they have a single shape. Like the Stella, all of them have star-like shapes. The Haloarcula, all of them are rectangles. But some bacteria have many different shapes, and these we call pleomorphic. Like here's a pleomorphic bacteria here, takes on many different shapes. One species. Any question about any of that? The bacteria can take on a cellular growth arrangement, also called the cell arrangement. That is when cells stick together, how they stick together. And when you look at them under the microscope, you can see a singlet, a single cocci here. Let me blow this up. A single cocci here, a diplococci here, where two cells are stuck together, and there's a diplococci there, right there. You can have a streptococci where the cells are stuck together in a, what do you call that, a row? And there's a streptococci right there. Excuse me. I warn you, I tend to sneeze twice. You can have a tetroid where four cells are stuck together, like there. They have two different divisions. The cell division, the cell divides on to make the tetrad. It's hard to see here, but there is a tetrad there separated from that tetrad there. And there's one there. What they needed to do was have fewer cells here so you could more easily see it. And then you have a sarxinia, which the cells divide on three different planes. And that's forming 8 cells here. And there's 3 there and 4 there. The fourth one is back there. And there's a sarxinia there. You can also have a clump of cells that we call staphylococci. And there it is there. If you put a stem on right there, this reminds people of a cluster or a clump of grapes. And staphylococci actually means ball-like cell in the shape of a cluster of grapes. That's what staphylococci means. Any question about any of that? The cocci have the most cell arrangements, meaning they can be singlets, diplococci, streptococci, tetrads, succinia, or staphylococci. The bacilli do have cell arrangements, but they have fewer of them than the cocci. The first, you can have a singlet, a single bacilli, shown here. You can have a diplobacilli, shown here, and there's actually a diplobacilli right there. 2 bacilli stuck together on this end, where they sort of are stuck together like a sausage link, where the cells are stuck together on their long ends. 2 of them. Streptobacilli is a row of them. In this case, 2 diplobacilli stuck together. And there we see a streptobacilli right there. 1, 2, 3, probably 4 cells there. If the cells are stuck together on the short side, we don't call them streptobacilli. We call them a palisade. Let me get paint open again. So the cells are stuck together this way. That's supposed to be straight. I didn't draw it real well. And if those of you have ever read Mark Twain, you remember that Tom Sawyer was painting a fence that was a palisade fence. And that's where the term comes from. And there we have a bunch of bacilli stuck together this way, what we call a palisade. And this is also a palisade fence, which is where you have the boards, one after the other, lengthwise. Any question about any of that? Those are the different cellular growth arrangements of bacilli, meaning only singlets, diplobacilli, streptobacilli, and palisade. A coccobacillus is not a cellular growth arrangement. It shouldn't be in this slide. It is the shape of the bacteria. And I mentioned it before. Let me show you that. What it is a bacillus, which has gotten shorter and shorter, and is rounded on the edge. And so it's in between a coccus, which that one actually looks coccus right here. Come on, mouse. That one actually looks coccus, but that one is coccobacillus. And that one almost a little longer, and it would be a bacilli, but it's short enough that we call it a coccobacillus. That's clearly a coccobacillus, and so is that one. Any question about that? You should note the term bacillus does have two different meanings. We have the shape, a rod, which is always a lowercase b, meaning the shape of the cell. And then we have the family, bacillus, which has a capital B. So be careful of the term bacillus. It does have two different meanings. The family bacilli with a capital B, and then the bacteria shape with a lowercase b. Let me go back and mention that be careful of streptococci. It is the cellular growth arrangement. We have the family of bacteria called Streptococcus. It's slightly different than streptococci, but this is usually lowercase s, and the family is always uppercase s, as well as the family is called Streptococcus, not streptococci. Any question about any of that? All right, if not, I'm going to end the lesson here, and I will see you at 6:30 if you have questions. If you don't have questions, you do not need to show up for the lab. Any questions? All right, I'll say goodbye and see you later. Guess I should leave that.